Chapter 12, Writing a Wrong Turn. Good and evil often sprout from the same tree, and the body is in Eden. Nothing illustrates this paradox better than cancer. Today, because of breakthroughs in genetics, thousands of scientists are searching for oncogenes, bits of DNA that are presumed to pull the trigger that fires the malignant bullet. It has been known for a long time, of course, that cancer isn't inherited through egg and sperm the way hemophilia is. However, many postulate that the immediate cause of cancer may be genetic changes in somatic cells. Normally suppressed genes held in an unnoticed corner of our genetic bookshelf since long ago in our evolution might be dusted off only when other bodily conditions are just wrong. While the premise of this idea is apparently true, biologists have recently concluded that the difference between a normal gene producing a normal protein and one that could theoretically cause cancer in a single typographical error in a whole chapter of amino acid sequences. Such mistakes happen so often that we would all be riddled with cancer from infancy that were all it took to start the disease. Something else must go awry before a few misspellings can turn the whole library into gibberish. 3 basic criteria by which a doctor diagnoses cancer must serve as the starting point in solving the mystery of its cause. First of all, the disease always arises not from the alien germ, but from a formerly normal cell of the host body. And the cancer cells are more primitive than their healthy precursors. Moreover, this atavism reflects the seriousness of the disease. The simpler the cells, the faster they grow and the harder they are to treat. Whereas a tumor that still resembles its tissue of origin is less malignant. The second criterion is growth rate. Cancer cells multiply wildly in contrast to the slow, carefully controlled mitosis of normal cells. Going hand in hand with this uncontrolled proliferation is a small lack of control in the structural arrangement of the cells. Their membranes don't line up in a normal, specific ways, and they form a jumbled mass instead of useful architecture. As a further result of runaway multiplication, cancer doesn't observe the boundary laws of normal tissue. Instead, it encroaches imperialistically upon its neighbors. In addition, since cells don't adhere in any kind of structure, some of them are constantly breaking off, flowing through the blood and lymph and setting up colonies, metastasis throughout the body. The third basic criterion of cancer is metabolic priority. The diseased tissues greedily takes first choice of all nutrients circulating in the blood. The healthy part of the body gets what's left over. As the tumors disseminate and grow, they consume all available food and the host wastes away and dies. We can make one crucial observation at this point. Except for the lack of control, all 3 characteristics, cell simplicity, mitotic speed, and metabolic priority, are hallmarks of 2 normal conditions, embryonic growth and regeneration. When considering the similarities between an embryo and a tumor, it's important to keep in mind one difference. Even though contained within the body of its mother, the embryo is a complete organism and the controls over its cells are primarily its own, not those of an adult. Over 30 years ago in Switzerland, G. Andres probed this relationship by implanting frog embryos in various body tissues of adult frogs. Whenever the host didn't simply reject the graft, the embryo degenerated into a highly malignant metastasizing tumor. As a result, Andres proposed a theory of cancer that remains provocative today. A normal cell becomes cancerous by dedifferentiation. The change is not dangerous per se, according to Andres, but because it occurs in a post-fetal animal, the controls that would normally hold these neoembryonic cells in check aren't working. Cancer's relationship to regeneration is even more interesting. In the latter, a rapid growth of primitive cells have metabolic priority occurs in adult, but with proper control as in an embryo. Those animals that regenerate best are least susceptible to cancer. In general, as complexity increases up the evolutionary ladder from worms to humans, regeneration decreases and cancer becomes more common. Although salamanders stand about midway in degree of complexity, they're perhaps the least specialized of all land vertebrates. They have tremendous regenerative abilities and almost no cancer. Even to give them tumors in the laboratory requires much effort. Adult frogs, on the other hand, have bodies that are much more specialized for their amphibious way of life. They regenerate very little and are subject to several kinds of cancer. As regeneration decreases, cancer increases. In 1948, Merrill Rose decided to see whether the environment of a salamander's regenerating limb could control the primitive cells of cancer, as well as those of blastema. He took pieces of type of a kidney tumor common in frogs and transplanted them to the limbs of salamanders. These tumors took better than most and soon killed the animals when allowed to spread unchecked. However, when Rose amputated the leg just below or through the malignancy, normal regeneration followed, and the cancer cells dedifferentiated more fully as the blastema formed. Then, as the new leg grew, the former frog tumor cells redifferentiated along with the blastema. The frog cells were easily distinguished from salamander cells by their smaller nuclei, and microscopic studies showed frog muscle mixed in with the salamander muscle, frog cartilage cells amid salamander cartilage, and so on. This monumentally important experiment proved Rose's hypothesis that regeneration's guidance system could control cancer too. It implied that cancer cells weren't special, but merely embryonic cells in a post-embryonic body. Rose's work led directly to Andre's theory a few years later. Rose's experiment. Unfortunately, biology was still firmly gripped by anti-dedifferentiationism, and these ideas were partially ridiculed, partly ignored. The reaction held back cancer research for decades because the dogma implied that carcinogenesis, like dedifferentiation, was irreversible. Once a cancer cell, always a cancer cell. As long as this view was sacred, the only possible way to cure cancer was to cut it out or kill it with drugs and x-rays. We've been beating that dead horse for 50 years now with tragically modest increases in survival rates. Surgery works only against tumors that haven't yet spread. Chemotherapy depends on differences between malignant and healthy cells. However, the differences aren't as great as we would like because cancer arises not in the same distant swamp, but from a slight change in our own cells. Therefore, chemotherapy and x-rays inevitably produce some damage in normal cells too. Doctors, being only human and sharing in their patients' pain and terror, have devised ever more ferocious treatments. In our war on cancer, we've stampeded ourselves into a sort of Vietnam syndrome. To destroy our foes, we're killing our friends. As Walt Kelly's cartoon character Pogo observed in that context, we have met the enemy and he is us. But surely you may be thinking, if this theory of cancer had any potential for a cure, the research establishment would have considered it. And surely there would be some supporting evidence. Unfortunately, even though the detooling and retooling of cells have now been accepted by all of biology, the old habits still persist through most of the granting hierarchy. A few years ago, for example, I met a young research fellow at National Cancer Institute who wanted to study the regeneration cancer link. He even showed me his proposal, an excellent one. I told him he was asking for trouble if he submitted it to NCI, but he said his boss approved and he was sure he'd get the grant. A month later, he was forced out of the institute, and the project has never been funded. Nevertheless, supporting evidence from research on the periphery does exist. Since regeneration can occur without the stimulus and control of nerves, one would expect them to exert some control effect on cancer. They apparently do. As far back as the 1920s, several experimenters implanted tumors into denervated areas. Without exception, the cancer cells took root better and grew faster than where the nerves were intact. The early work on this point was criticized on the grounds that denervation might have reduced the efficiency of the circulatory system, which in turn would have enhanced malignant growth. Then in the mid-1950s and 60s, more sophisticated techniques established the same relationship. Absence of the nerves accelerated tumor growth, and the variations in the blood supply had no significant effect. Further evidence confirming Rose's conclusion that regenerative controls cause tumors to regress came from a series of experiments by F. Saylern, Aspang, and K. Kratochwil of an Austrian Cancer Research Institute in 1962 and 1963. They worked on salamanders, but instead of implanting frog tumor cells, they induced skin cancer with large repeated applications of carcinogenic chemicals. With persistence, they eventually got tumors that would invade subsurface tissue, metastasize, and kill the animals. In one series, they applied the carcinogen to the base of the tail. The primary tumor formed there, and the metastasized appeared at random in the rest of the body. If they then amputated the tail, leaving the primary tumor intact, this malignancy would disappear as the tail regrew. Cell studies showed that it didn't die or regenerate, but apparently reverted to normal skin. Furthermore, all the secondary tumors vanished too, as though they were being operated by remote control from the main one. The salamander ended up with a new tail and no cancer. However, if the primary tumor was at a distant point on the body, amputation of the tail had no effect. Even though the tail regenerated, the main cancer and its offshoots all progressed and the animal died. This research, combined with Rose's, indicates that regeneration near a primary tumor can make it regress by reverting to its normal tissue type. I doubt that there's anything special about legs or tails. I would predict that regrowth in any part of the body, as long as it was near the primary tumor, would have the same effect. The key to regression appears to be a change in the malignancy's immediate neighborhood. The electrical currents in the nerve, and particularly in the neuroepidermal junction, seem likely candidates, since they surface to start regeneration in animals normally incapable of it. There's abundant evidence that the state of the entire nervous system can affect change. Back in 1927, Alida Evans, a student of Carl Jung, documented a link between depression and cancer in a study almost totally neglected in the intervening years. And a long-term project began in 1946 by the Carolyn Bedell Thomas at Johns Hopkins School of Medicine. Students were given personality tests, and the occurrence of disease among them was charted over several decades. In this and later studies, a high risk of developing cancer has been correlated with a specific psychological profile that includes a poor relationship with parents, self-pity, self-depreciation, passivity, a compulsive need to please, and above all, an inability to rise from depression after some traumatic event, such as the death of a loved one or a loss of a job. In such a person, cancer typically follows a loss in a year or two. Several physicians have found they can greatly increase cancer patients' chances of a cure with biofeedback, meditation, hypnosis, or visualization techniques. Several years ago, Carl Simonton, an oncologist, and Stephanie Mathis Simonton, a psychologist, began using all these methods with emphasis on having patients develop a clear picture of the cancer and their body's response to it. For example, a patient might spend a meditative period each day imagining white blood cells as knights on white horses defeating an army of black-caped marauders. When the Simontons tabulated their first results in normal terminal cases, they found that 159 people expected to die in less than a year. Those who eventually did succumb lived twice as long. The cancer had completely regressed in 22% and was receding in an additional 19%. These results have held up, and visualization is now being adopted in some other cancer treatment programs. Penn State psychologist Howard Hall, testing hypothesis for boosting white cell activity, found 40% increase in cell counts among this younger, more responsive subjects just 1 week after the trained session. New Haven surgeon Bernie Siegel has developed the Simontons methods further with therapy groups called experimental cancer patients. Having patients make drawings to reveal their true psychological state, sans defenses, then working towards a comprehensive change in outlook, total CNS response, to mobilize the will to live. Siegel has helped his patients greatly improve the quality and quantity of their lives as compared to clinical prognoses. This approach also enhances chemotherapy's effectiveness while minimizing its side effects and dramatically increases the likelihood of a miracle, total regression, and a cure of cancer. All of this shouldn't be so surprising. Under hypnosis, the mind can completely block pain, and research described in the next chapter has shown that it does so by changing electrical potentials in the body. How can we be sure it couldn't create appropriate electrical changes around a tumor and melt it away? There are still major problems with these psychological approaches, however. Only a minority of people are able or willing to muster the high level of dedication needed to make them work, even under the gun of a death. Moreover, they require time, the very thing a cancer patient is short of, and they often don't produce a complete cure, even when they practice diligently. Still, they're encouraging signs that the tide is beginning to turn from the warfare mode to the simpler, more elegant mathematician would say, idea of changing the cellular environment that allows a tumor to flourish. Could we really do the job more directly by applying the proper electrical input to aim a surefire regenerative influence at the tumor? I'm sad to say that most of the few researchers who've tried electricity against cancer have used the kill the enemy approach. Tumors are somewhat more sensitive to heat than normal tissue. So some doctors are using directed beams of microwaves to cook them without its hope destroying too many healthy cells. FDA approved for general use of this method is expected soon. It has been known since the time of Burr and Lund that growing tissue is electrically negative, and cancer is the most negative of all. Hence, some researchers have tried to inhibit tumor growth by canceling the offending polarity with positive current. Early reports were encouraging, but we know what that toxic metallic ions are released from most positive electrodes. So this method must be tested with great caution. Only one research team has sought a integrative effect of electric current. In the late 1950s, Carol E. Humphrey and E. H. Still of the Applied Physics Laboratory at Johns Hopkins tried pulse direct currents on standardized fast-growing skin tumors in mice. Even though they used both positive and negative polarities, their results seemed sensational. In one series, they got total remission in 60% of the test animals after only 3 weeks. All the control mice had died by then. In another series, the control tumors averaged 7 times the size of ones treated with current. Unfortunately, present evidence doesn't really support this approach either. In my group's experiments with human fibrosarcoma cells in vitro, negative and positive currents both speeded up growth by over 300%. On the other hand, as mentioned in Chapter 8, we found that we could suspend mitosis in fibrosarcoma cells with silver ions injected by minute levels of positive current. During one day of exposure, the cells appeared to dedifferentiate completely and they stopped dividing for a month without additional treatment, even though we changed the nutrient medium regularly. Obviously, this entire subject needs to be investigated more thoroughly. Some researchers believe pulse electromagnetic fields may be some of the benefit in treating cancer. Art Pillow, working with the Larry Norton and Lori Transman of New York's Mount Sinai School of Medicine, as well as William Regelson of the Medical College of Virginia, claims to have found a pulse sequence that significantly increases the survival time of mice with cancer. So far, these experiments say they've increased the effectiveness of chemotherapy in lab animals with PEMF, but haven't found a pulse pattern that consistently regresses tumors in vivo. Although Pilla and Steve Smith have been able to transform malignant lymph node cancer cells into benign fibroblasts in culture. The claim that PEMF may retard cancer in animals is seriously flawed, however. In these experiments, the entire animal was exposed to the field, not just the part with cancer. The pulsing field, like almost any time varying magnetic field, induces a stress response in the animal. For a short while, this increases the activity of the immune system, which slows the growth of the tumor. However, the field's effect on the tumor itself is to speed up, and in the long run, added stress is the last thing an animal with cancer needs. These experiments cannot be used to indicate the safety or benefit of PEMF in regard to cancer. Since to heal bones, the fields are directly only at small regions, PEMF, as used on humans, does not produce stress, increased immune response, or any concomitant anti-tumor activity. Certain leads, such as the electrically injected silver, remain promising. In the 1950s and 60s, Dr. Kenneth McLean published some interesting work on the use of magnetic fields versus tumors in mice. He believed he'd healed several cases of cancer with steady state magnetic fields, and certain unorthodox healers in America and India who use permanent magnets have made similar claims. The difference in effect between steady state and time varying fields leads me to theorize that a steady state magnetic field, if strong enough, may indeed halt mitosis in malignant cells. Due to the prevailing outlook in cancer research, the key work remains to be done. Even promising non-electromagnetic approaches have been victims of bias. There's sound evidence, for example, that megadoses of vitamin C do slow tumor growth and increase the chances for a complete cure. But Linus Pauling hasn't been able to persuade any of the powerful institutes to perform a large-scale trial. Some animal tests are now being funded, but since vitamin C is non-toxic, immediate clinical experiments on large numbers of humans would make much more sense. And following up the regeneration connection, two experiments in particular are crying out to be tried. Someone must attempt to duplicate the electrical environment of regeneration around tumors in lab animals using electrodes. This would involve introducing small negative currents to thoroughly test the hypothesis that cancer cells are stuck in the state of incomplete dedifferentiation. The idea would be to dedifferentiate them the rest of the way and then let normal processes in the body turn them into healthy, mature cells. Same hypothesis should be tested another way by surgically creating neuroepidermal junctions near the tumors. Will these experiments be done soon? I wish I knew. The multi-billion-dollar cancer research bureaucracy could certainly afford them. But although there are a few signs of change, the establishment is stuck in the near primitive state of war mentally. I've maintained for many years that we won't learn much more about abnormal growth until we learn more about the normal kind. That approach can lead to cancer treatments that are truly compatible with our bodies, far safer and more effective than the simplistic, dangerous ones now in vogue.